having me. It's a pleasure to be here. So what interests me most in plant research is the relationship between the environment and the molecular and physiological response. So cotton has my research interest at the moment. I work with Dr. Hong Zhang. I don't know, some of you may know him here. So why is climate change important to this region? It's important because we see a lot of drought, and drought is the major cause of crop loss throughout the world, not just here in Texas, but worldwide. So it's a consistent problem. So where does our water come from that we use for agricultural production? Here in West Texas, it comes from the Ogallala Aquifer, but studies have shown that the water's being depleted. So we really have a limited water supply. So in the future, our demands on agriculture are really going to rely on our dry land instead of irrigated crops. So that's kind of what our research focuses on, is what are we going to do in future? So that technology is in our hands and we can focus on that. And also, whenever it dries up, you can see poor water quality, so there's a salt issue. So we also look not just at drought, but at salinity issues. And then you never see drought stress coming just one stress factor at a time. You always see it go hand in hand with things like heat stress. So heat stress is another factor we look at. So what happens to a plant when it's under high thermal stress? Like for cotton, the optimal temperature is probably around 84, 86 Fahrenheit, 28 Celsius, somewhere in there. But we never see that in the summer. It's always much more warm. So that's part of the climate change. So that's a real challenge. So what we see with plants is you see enzymes and proteins being degraded. You also see membrane fluidity issues. And then from my standpoint, we see photosynthesis just ceases to cycle. That's a problem for the plant. When the sunlight energy can no longer turn into chemical energy for the plant, it's a problem. So that's some of the things we're looking at for our research for drought, heat, and salt tolerance. So as a scientist, we have many questions. So as a molecular approach, what are some of the things we look at? We look at what biological functions most affect the plant. What pathways are involved? What genes? What proteins? What can we do to aid the plant to better tolerate these harsh conditions? So that's part of what our lab does. But the important thing is a lot of these genes that we look at, they're already inside the plant. So it's not some kind of mutant we're looking at, it's already there. So for the drought example, the limiting factor is osmotic regulation. What can we do to aid the plant for osmotic regulation? So we're looking at a proton pump found on the vacuole membrane, and by overexpressing the gene for this proton pump, now we have more protons that can be pumped in, not just for osmotic regulation, but it also can affect other factors of the plant. For salt stress, for example. So now the sodium proton antiporter, for every proton being pumped out, you can pump in a sodium proton, sequestering it there where it's safe in the vacuole and making the cytoplasm less toxic. So now by overexpressing one gene, we've created tolerance to drought and salt. Well, now we go back to the heat issue. What about photosynthesis? What inside the photosynthesis system is the rate limiting step? I see that as the Rubisco enzyme. So how do you reactivate an enzyme, especially in photosynthesis? You need energy. You need ATP energy. So it's a Rubisco activase. So by overexpressing a Rubisco activase, a more thermostable Rubisco activase, you're going to see a higher catalytic turnover, higher photosynthetic rates during times of stress. So now the plant has that chemical energy to yield higher under stress. So that's some of the things. We take both these genes, co-overexpress them in cotton, and now we can study it. So that's kind of the point I'm at in my research, is taking these overexpressed plants, comparing them to wild types, using chambers to check for heat and drought, using the greenhouse for salt and drought. We have field conditions under dry land, and we do see an increase in photosynthetic rates with the overexpressed plants compared to wild type. We see yield increases, and it's not just my research. I also have a colleague that has promising results with her research as well with this technology. So it's there in our hands for the future of agriculture. We have the technology, we just have to use it. So it can be sustainable. But why is this important to our region? Because cotton is the #1 cash crop here in Texas. So it is important. So I just wanted to leave you with our story, and you can follow us along in our website and continue to see how we do.